So, this week we'll be discussing nucleic acids, RNA, and protein synthesis, along with basics of molecular biology. Last week we have already discussed enzymes as drug targets, enzyme kinetics, and inhibition, and various modes of inhibition. So, we won't be doing that again this week. Let's start with the basic introduction to molecular biology. That's with nucleic acid. Now, why are RNA and DNA called nucleic acid? Well, I'm audible to you guys, right? Just give me a thumbs up or anything, and I, perfect. Thank you. So, why are RNA and DNA called nucleic acids? They are not acids. They don't have carboxylic acid, the like conventional acids that you think of. Actually, as we all know, RNA and DNA are both found in nucleus of a cell, and they have hydrogen, nitrogen, carbon, and phosphorus and oxygen group present in it. And due to the presence of phosphate group, it has acidic nature, and therefore it's called nucleic acid. Now, DNA like RNA, DNA like protein has primary, secondary, and tertiary structure. It's different in the sense that it has different bond and everything, but we'll talk about it now. So let's discuss the primary structure first. The primary structure, I'll discuss this very briefly because you have already gone through the weekly lessons. This is just to help you recapitulate all the concepts and to solve the questions. So the primary structure consists of a building block called nucleoside that has two components, sugar and base. Sugar is deoxyribose sugar, and that's same for all nucleosides. The only change that makes a nucleoside is change in the base pairs. Now, this base pair can be purine and pyrimidine. Like a purine, say adenine, guanine, and pyrimidine is thymine and cytosine. A nucleoside with sugar is called, as you can see in the screen, deoxyadenosine when it has adenine attached to it, guanosine when guanine is attached to it, thymidine when with and vice versa. So when phosphate group is present in it, it's called nucleotide. And basically, now the joining of this bond is through phosphodiester bond. You can see here this bond, and this bond is what joins all these building blocks of nucleoside together, and they form the primary structure of DNA. Let's solve a question and see what it is. So, which of the following best describes the primary structure of DNA? A double helix formed by complementary base pairing. B, the linear sequence of nucleotide linked by phosphodiester bonds. C, the interaction of DNA with histone proteins. And D, the supercoiled structure of DNA in chromosomes. Please put your answers in the chat box so that I can know if you're clear with the concept. Very good, I can already see the answer. The one thing about DNA that I forgot to mention while going through it is it always has this complementary base pairing thing. So adenine, A will always form bond with T, and G will always form bond with C. So ATGC. Hence, every time there, it is a double helical structure. So what happens is every time there, and the number of purine is always equal to number of pyrimidine. So answer for this question is B. The primary structure of DNA refers to the linear structure of nucleotides joined by phosphodiester bonds, and that is how it encodes all the genetic information. Second question. Which of the following statement about the primary structure of DNA is false? A. The nucleotide sequence is written in 5 to 3 direction. B. Is B. It consists of deoxyribonucleotides linked by phosphodiester bonds. C. The bases are attached to the phosphate backbone by peptide bonds. D. The identity and order of nucleotides determine the genetic information stored. Yes, I think everyone is right. The answer is C. So we know that the nucleotides are always written in 5 to 3 direction. Let me go to the first slide again. You can see in the first, in the second structure, this structure, the 5 end has free phosphate end, whereas the 3 structure has free hydroxyl group. Now, whenever we read about this base pair and everything, just wait a minute. I don't know what's happening. So, whenever we read the sequences, we read from 5 end to 3 end. And obviously we discussed about the phosphodiester bond, so we know that the bonds between this primary structure is because of the phosphodiester bond. The bases are attached to the phosphate bond by peptide bonds. No, actually that's not correct. The primary structure of protein has this peptide bond, CONH bond. This peptide bond is not present in DNA. The bonds that are present, you can see here, is the bond between sugar and the base, as you can see here, is N glycosidic bond, the N from the base, and the bond from the sugar, this is called N glycosidic bond. And the other bond that we see is this phosphodiester bond, this bond. So obviously the third one is wrong. The identity and order of nucleotides determine the genetic information stored. Well, this is a correct answer because the sequence of DNA, they determine the codons, and the codons code for the amino acids, and this amino acids form the protein structure. So they are, the sequence of nucleotides determine how the information travels from DNA to protein. Let's briefly discuss the secondary structure of DNA. Now, secondary structure of DNA, as you all know, is a double helical structure. This was determined by Watson and Crick based on data produced by, Rosalind Franklin. That's another discussion. But so we know that there's a rule of complementary base pairing between the different bases, between the 4 bases that we have in DNA. So A, T, G, C. A has A always forms a bond with T, and it has two hydrogen bonds there, whereas there are 3 hydrogen bonds between G and C. You can see it from here. You can see C here forms bond with G, and there are 3 hydrogen bonds, 1, 2, and 3. Whereas with the bond between the hydrogen bond between A and T is 1 and 2. This basically forms a ladder like structure between these two, and that's how they interact. Apart from this, you can see the structure of purine and pyrimidine. They have this cyclic structure unsaturated cyclic structure. So what happens is they have this pi-pi interaction among themselves. Upon this bond formation, they also have this pi-pi stacking, that's hydrophobic interaction between each other. This creates some kind of like, torsion among the ladder-like structure that DNA has, and hence it forms like, this double helical structure based on these two forces, like stair-like structure because of the base pair, complementary base pair, and then because of the hydrophobic interaction between the stacking of the base pair, they form this double helical structure. Now, how, you know let's discuss how this is basically formed. We'll discuss more about it, in the upcoming slides. But just remember, whenever there is like, addition of base pair, it happens from 5 dash end to 3 dash end. So suppose there is one, the primary structure of DNA. It has like one strand. Now one strand here, you can see this from 5 dash n to 3 dash n. We see this is G and C. So GC, they have three hydrogen bonds with their complementary base pair. And so now you will see there is another base pair, that's T. Now T will form bond with A. What happens is a nucleotide comes to form bond with T. Obviously this nucleotide is A because it's a complementary base of T. It comes, it approaches T, and there it forms like double hydrogen bond with T. And what happens is after that the phosphate group leaves. We can see the mechanism here. See, it was already GC. This is the template from 5 dash n to 3 dash n. Don't worry, I'll be more elaborate while discussing replication. This is just for a general understanding. Now you can see, um, this is the nucleotide, and this nucleotide again, this adenosine. So deoxyriboadenosine is trying to approach that, it's forming this hydrogen bond, and the free hydroxyl group, again the 3 dash end of the sugar, deoxyribose sugar, it has, the lone pair. This will attack the phosphate group, and which in turn will break this phosphodiester bond, and this group will leave, and hence the bisphosphate group leaves, forming this phosphodiester bond between the two nucleotides. This is the phosphodiester bond that forms the, backbone of the DNA. Hence we get the secondary structure of DNA, that is double strand, that makes the DNA double stranded. Now let's try to solve few questions on secondary structure of DNA. The secondary structure of DNA is stabilized primarily by A. Hydrogen bonds and hydrophobic interactions, B. Covalent disulfide bonds, C. Peptide bonds, D. Van der Waals forces. Perfect. Yes, you guys are right. It is actually, indeed, you, yeah, perfect. Good. So the correct answer is A. Hydrogen bond between this base pairs and hydrophobic interaction between the base pairs due to after their bond formation among each other. So this altogether stabilizes the secondary structure of the DNA. Now, next question. What is the primary force that drives, this is interesting question. See, what is the primary force that drives the formation of the double helix structure of DNA? A. Hydrogen bonding between complementary base pairs. B. Covalent bonding between sugar phosphate backbone. C. Hydrophobic interaction between nitrogenous bases. And D. Electrostatic interaction between phosphate groups. The important thing to notice here is the primary force that drives the formation of double helix structure. So, if there was no pi stacking, it could have been in form of like a ladder structure, right? Three base pair, three hydrogen bond with the complementary base pair, two hydrogen. It could have been like a gradual stair-like structure. Why does it become double helix? So the correct answer here is actually C. Though the hydrogen bond stabilizes the helical structure, the driving force between the double helix structure is hydrophobic interaction between the base pairs. As they stack among themselves, you know, yeah, they try to um, you know try to minimize their surface area to avoid hydrolysis, and they come in a torsional state, like in a double helical structure due to the interaction, due to the pi stacking of due to the hydrophobic interaction from the pi stacking of the nitrogenous bases. I hope this is clear. Would you like me to repeat this again or discuss the secondary structure again? Please let me know. You can unmute yourself and ask me question if you have any. Otherwise, I'll move to the next topic. Very well. So now let's discuss the tertiary structure of DNA. So now we know that DNA is very, very long, and there's certain parts in DNA that do not even have like genetic material. They are like repeating units that do not code for any kind of protein or anything. So that makes the DNA very long, you know, very long thing. So now, but we know that DNA exists in, nucleus, let alone the cell. So how does the DNA, pack itself to fit in the nucleus of the cell? For the folding of DNA, just like in case of protein folded itself in a tertiary structure. DNA also folds itself in a tertiary structure. Now, secondary structure was double helical structure. So you can imagine it's already, not very easy to coil DNA in tertiary structure. What happens is, you know, just take the example of this rubber band. So you can see this, don't mind, please assume that it's just a plain structure. So this is the helical structure, right? The helical structure of DNA. To fold it, it has to, and to make a compact structure, it has to fold in certain way, like it goes inside each other, right? They have to like be very compact, and that happens with the help of a class of enzymes known as topoisomerase. They have like there are more different kinds of topoisomerases as well, like topoisomerase 1, 2, 4, and in E. coli 2, 3 as well. So we'll not discuss about E. coli right now, but in general, uh, there are two topoisomerases involved in supercoiling of DNA, and the supercoiled structure is called tertiary structure. Now, the tertiary structure, to make the tertiary structure, what topoisomerase does is topoisomerase 2, it basically nicks DNA, the two strands of DNA at different places. After nicking it, it just it gives it's like more free energy to rotate and to crumble and form the tertiary structure. As you can see here, topoisomerase 2 has free tyrosine, and this topoisomerase it just comes and nicks the double stranded DNA. This place as well as in this place, you can see it cleaves the DNA, the strands of DNA at two different positions, and gives it certain kind of flexibility to fold. Now once it comes to both the position, and after folding it just leaves it, and reattaches, reseals the DNA. You can see the organic chemistry behind it here. This is topoisomerase 2. It has free tyrosine. We know tyrosine has OH, no CH2. This tyrosine, tyrosine here, it attacks again. The OH attacks the phosphate bond, PO bond. It attacks the phosphate, and then there is hydrolysis of this bond, and we get OH free, and then again the it forms PO bond with the tyrosine, and that's basically phosphorylation of the tyrosine in topoisomerase 2. This is how it splits and or cleaves the DNA for supercoiling. The DNA, basically the tertiary structure in it, what happens is, you can see here, this is the double stranded DNA, and this is the secondary structure. It forms itself. There's a structure called histone, and the DNA coils around itself, and not itself, coils itself around the histone protein, and that altogether, the histone protein along with the coiled DNA is called nucleosome. Now this nucleosome, again you can see here, this is the nucleosome, and these are also nucleosomes, right? These nucleosomes are together, held by spacer DNAs. These are called spacer DNAs that are not exactly folded in nucleosomes, and there are many nucleosomes together make form, they lie together and they make chromatin, and they are further folded in nucleus, you can see here. So that's the tertiary structure of DNA. That's clear, right? Would you like me to repeat this again? Yes, sir. Sorry. I'm Yeah, I could say like yeah, that's um chromatins. I would say that would be included in the tertiary structure of DNA. You mean can you be more elaborate with your question? I'm sorry, I'm getting confused. Can you be a little more elaborate with your question? Yeah. Yeah. To form nucleosome. Yeah. Yeah, they all together form, yeah, yeah, yeah. Along with spacer DNAs, they form chromatins. Yeah, so, yes, yeah, tertiary, yeah, till that point, we can call it tertiary structure. And in some cases, there's quaternary folding as well, but um, that still, there it's like further packing, and that's not generally dealt with at this level. So yeah, till that point, it's tertiary structure. I thought there's further folding of chromatin. Thank you. It was really good question. Thank you. If that's all, I'll move ahead. Now let's briefly discuss what replication is. Replication is production of two identical replicas of a DNA from original DNA. That's basically uh, copying the genetic information from one generation to the next generation. Now, as you know, that DNA is a double helical structure. So, in order to make two pairs of DNA, it has to uncoil, and one of the strand will code for one strand, and the other one will code for another strand. So, one DNA strand will altogether make two copies of DNA. Now, let's see what that happens. Now, the structure, as you can imagine, is, you know, uh, firmly held together. Now, first we have to unwind that, and this is where helicase enzyme comes. Helicase unwinds the DNA and breaks it open. Once the DNA is opened, the next enzyme comes, that's called single strand binding protein. So once a DNA opens, it has a tendency, it wants to go back to its original position. Like, the H bonds want to be formed, you want to be back again, like, form double and triple bonds again. In order to keep the helix open, these proteins come. These proteins, they stabilize the unwound parental DNA, and then DNA polymerase starts, basically making the bringing the complementary base pairs from 5 dash direction to 3 dash direction. So you can see here, this is the parental DNA, this is the template DNA strand. Once it opens, like, there is 5 dash to 3 dash, and the complementary would be 3 dash to 5 dash. The 3 dash end to 5 dash end is called parental DNA. It's not, you know uh, it's not the one that we take in reference for. The first one that we are taking in reference for is like 5 dash to 3 dash. Now, the uh, the replication takes place in the complementary, in the opposite direction, that is, sorry, that is from 5 dash of the unwound DNA to 3 dash end. You can see here, this is how DNA polymerase starts working. So it starts putting on, like, bringing the complementary base pairs and starts stitching, like zipping the DNA. The fourth one, now let's come to the long one. This one is where RNA primer, this is where primase comes in. It starts making DNA. uh, so this is not happening in a continuous manner. This is called, this takes place in fragments, bits and parts, and it's called lagging strand. And once this is around, this is once this happens, DNA ligase comes back, and it starts to join the fragmented portion of it again, and we end up with two fragments of, two strands of DNA. You can see ligase, primase, polymerase, SSP, helicase. These are the 5 enzymes that are involved in the replication of DNA. Now let's get to the questions and test out our concepts. If a DNA template strand has a sequence 5 TAC to 3 N, what would be the complementary sequence in the newly synthesized strand? A, 5 ATG, that ends with 3 3 ATG, 5 and from 3 to 5 TAC, and D, 5 to 3 TGC. Good, I can already see the answers coming. So, yes, this was the sequence we were given. Complementary to C would be J, A, T, and T, A. And because they are complement, this one would be 3 dash to 5 dash. So this is what our answer is. So the correct answer is obviously which was the correct answer? 83 to 5. That's B. So because again the complementary nature of DNA and everything. So would you like me to explain this answer or should we just move forward? Okay, perfect. So this again explains the just employs the concept of complementarity of bases. Let's go to the next question. During DNA replication, a trinucleotide sequence 5 dash G 523 dash GTC in the template strand will result in the incorporation of which sequence in the newly synthesized strand? Okay. Again, 5 dash to 3 dash GTC. So this one would be 3 dash to 5 dash C G A C. Everyone is correct. It is the answer is B. Same concept. Very good. Everyone is okay. Now let's go to the next question. Which of the following correctly describes the function of DNA polymerase 3 in prokaryotic replication? A. It synthesizes RNA primer to initiate replication. B. It removes RNA primer and replaces them with DNA. C. It elongates the leading strand continuously and the lagging strand in fragments. D. It unwinds the DNA double helix. So the unwinding work is done by helicase enzyme. So no, DNA polymerase is not involved in unwinding of DNA double helix. Um, DNA polymerase, you know, let me go to the replication. You can see the DNA polymerase, it basically brings, all the complementary DNA, it forms another unit. So basically, another strand of DNA, and then it comes back to Okazaki fragment. And here also, it starts to join the discontinuous uh, fragments of DNA that were born, because it does not have that kind of energy. RNA primer does not have that energy to synthesize continuous strand of DNA. That is something that DNA polymerase does. So you can see, yeah, sorry. Yeah, and it does not read because that's one reason, and also it's very energy expensive process, and it puts all its energy. Yeah, exactly, you're right, that's the correct answer. But also yeah, that's right. So the correct answer here is C. It elongates the leading strand continuously and lagging strands in the in the fragments, like in Okazaki fragments. Does anyone have doubt with this question? Now let's go to the next question. Which enzyme is responsible for unwinding of DNA helix during replication? A. DNA ligase, B. DNA polymerase, C. Helicase, D. Primase. Yeah, everyone is correct. The right answer is helicase enzyme. So let's go to the next question. Which enzyme is responsible for synthesizing an RNA primer during DNA replication? Yes, perfect. We know DNA polymerase, uh, you know, synthesizes the complementary DNA strand. Topoisomerase is involved in cleaving and supercoiling. Helicase is responsible for unwinding the double. Yeah. The perfect. The correct answer is primase. Let's move on to the next question. Which enzyme release supercoiling ahead of replication fork? A. DNA helicase, B. DNA ligase, C. DNA gyrase, which is also topoisomerase 2, and D. Primase. Well, that's a correct. Everyone is correct. The correct answer. Yeah. C is the correct answer. DNA gyrase, topoisomerase, that basically cleaves the DNA structure for to help the supercoiling process. Now, topoisomerase one, there are four topoisomerases: topoisomerase 1, 2, 3, and 4. 3, we don't talk much about 3 because it's present in E. coli, special, few of the E. coli, not even all E. coli, but in general, topoisomerase 1, it cleaves one of the strands of DNA, while topoisomerase 2, it cleaves both the strands of DNA for the supercoiling thing. Topoisomerase 4 is another bacterial enzyme that carries out the same process and is, important target for fluoroquinolone antibacterials. The next question is, which enzyme is used to cut DNA at specific sequence during recombinant DNA technology? Yeah, the correct answer is restriction endonuclease. It's also called molecular scissors because it cuts DNA in a particular sequence. After a particular sequence, it recognizes the sequence and it cuts the DNA there. So, yeah, they're used in recombinant DNA technology. We'll read more about it in the upcoming lessons. Let's move on to the next question. Which enzyme is a common target for antibiotics in bacterial DNA replication? Yeah, I should have written here. Let me add this here. DNA gyrase is also called topoisomerase 2. So this would help. Yes, The correct answer is DNA kinase, that is topoisomerase 2. There's a class of, in the previous question, I think it was there somewhere. Fluoroquinoline. They are a special class of antibiotics. They basically target topoisomerase 2 to basically stop the supercoiling, and we'll talk about it more when we start discussing different classes of antibacterial agents. So at this point, just remember this fluoroquinolones target specifically topoisomerase 2. Let's move on to the next question. Genetic polymorphisms are best defined as A. Single mutation causing genetic disorders. B. Variation in DNA sequence that occur in at least 1% of the population. C. Changes in chromosome number. D. Mutation induced by environmental factors. Now, genetic polymorphism. So, polymorphism, poly would stand for many, morphism is basically variation. So, yes, I'll wait for you guys to answer. Good. I can already see people answering the correct answer. So the correct answer is actually B, variation in DNA sequence that occur in at least 1% of the population. So let's briefly discuss what this genetic polymorphism is. Genetic polymorphism are common variations in DNA sequence. You know, again, if more than more, it's like more than one or more variants are, but is present in a particular population in the same locus of the gene. So it has to have at least two variants, and it's caused due to mutation. Now there is difference between mutation and polymorphism. Mutation can be in less than 1%, or maybe 0.5, 0.2% of the frequency of mutation can be very less, but in order uh, for it to be polymorphism, it has to be in a population where out of 100 at least one person has to have that variation, that genetic variation in his genes. So you can see presence of two or more variants forms oh yeah presence of two or more variant forms of a specific DNA sequence that occur among different individual in a population. It's a population attribute. That's the main thing. A gene is said to be polymorphic if more than one allele occupies gene locus within a population. Again, that's true, and it happens due to mutation. This is a population attribute, please remember that, and it has to be more than 1% in a population to be called polymorphism. It should be hereditary, and it should not in any way interfere the reproduction capability of a population. That's how it travels from one generation to the next. And if it's heritable, suppose it's heritable and its advantages to a population, it travels from one generation to other. And if it's not, if it's a disadvantage, we say advantage, disadvantage in terms of reproductive fitness. So if it's of any kind of, if it proves of some kind of a disadvantage for the population, it stops there. It does not move from one generation to the next. Now there can be different types of polymorphism: single nucleotide polymorphism, SNPs, and the next one is insertion or deletion of polymorphism, polymorphic repetitive sequence. The third one is CNV, structural or copy number variation. We'll discuss all this very briefly. So, you know, um, in single nucleotide variation, there is just change in one of the base pair. Suppose it's A, T, G, C, T, A. The variation would be A forms T, A, G, C. I'm just, yes, I'll definitely share this PPT. Now uh, the different sequence, the other sequence would be, suppose there is just mutation in A, instead of A or T, it'll become C. Rest all of them would remain the same. So just changing one single nucleotide is called single nucleotide polymorphism. It's present in, say, 1 out of every 100 individual. The deletion and insertion polymorphism, it's a specific nucleotide sequence of various lengths that is either inserted or deleted, and it's widely spread. Now, polymorphic repetitive sequence, it's very number of tandem repeats. Now, tandem repeats are, say, T A T, just wait. Let's say T. You can see a pattern like this is a repeating unit of the sequence of nucleotides. So this is called tandem repeats. They keep on repeating themselves. Now, this can be in centromeres or telomeres, and they comprise a large amount of tandem repeats. Depending upon how many base pairs are repeating, they can be macro, mini, and micro. For it to be a macro, the repeats has to be more than 100 base pairs. For mini, it's less than 50 base pairs, and for micro, it's less than 10 base pairs of tandem repeats. And a copy, the last one is copy number variations. It's also called structural because the entire allele is either repeated or you know deleted, so duplicated or repeated. So it just changes the entire structure of the allele. So it's called copy number variation. But what's the significance of this polymorphism? So you can see we have like there are certain things that are common in all entire population. Suppose everyone has insulin, right? But they have different way of their different way in which the insulin is regulated. Someone has better efficiency. Some people are insulin resistant, but that's again, I'm not going to talk about type 1 diabetes, type 2 diabetes, but in general, insulin regulation. Color of our skin, we all have melanin, but there is some people have more expression of alanine and melanin, sorry, and they have different color. The complexion is a very, it's varied even in a population. So exactly. So these polymorphism is the main. This is because of polymorphism, the way different genes are related and how they interact. There are certain factors like sexual polymorphism is male and female. They're just two types. Oh, that's controversial again, but genetically speaking, Yeah, let me scratch that. Let's not talk about that. Take example of like blood. Um, there's polymorphism in blood as well. There are different groups, A, B, O, A, B, O. You know, so yeah, that's where polymorphism comes. Now let's briefly discuss the difference between DNA and RNA. DNA is double stranded. You can see here RNA single stranded. DNA has different sugar, you know, deoxyribose, whereas RNA has ribose. You can see difference in the sugar as well. This, sorry, this has additional hydroxyl group compared to deoxyribose. And instead of thymine, which we see like in DNA, RNA has uracil. Uracil, you can compare the structure of thymine and uracil. You can see there is no methyl group in uracil. There is one in thymine. It just makes it more stable, and thymine is more stable compared to uracil. I'm not so okay. So that's basically RNA. RNA, like DNA, also has a primary structure and secondary structure. Secondary structure, it's just single stranded structure of RNA. RNA has three main types. We'll discuss more about illness in upcoming slides. Okay. So there are three main types of RNA: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA. That's called rRNA. mRNA carries the genetic information required for the synthesis of protein from the nucleus to endoplasmic reticulum. The rRNA is the main constituent of ribosome where protein synthesis takes place. The protein synthesis take like translation. It basically, happens in our ribosome. So smaller subunit and larger subunit. We can we'll discuss more about it in the next slide. So rRNA, a ribosome moves along mRNA, and that's how they code. tRNA interprets the coded message in mRNA. It contains the anticodons of the three nucleic bases which binds to the complementary triplet of mRNA. Each tRNA carries a specific amino acid, the nature of which is determined by anticodons. Um, each amino acids are coded by triplets. You know, three pairs of nitrogenous bases. They code for a particular amino acid. So we'll talk about it more in translation and transcription. And now there are three main types of RNA. But there are few more types of RNA when we go more to it. So yes, take example of like just remember this. We'll not discuss this. In a deeper. Will not consider. Just remember this. Okay. mRNA, messenger RNA, it's involved in translation, that's in protein synthesis. tRNA is also involved in translation, and rRNA, all three of them are involved in translation. The hnRNA, that's called heterogeneous nuclear RNA. It's precursor of mRNA and other RNAs. Like before they become RNA, they are hnRNA, and scRNA, they are no, small cytoplasmic RNA. They are involved in signal recognition particles and have like catalytic purpose. snRNA, small nuclear RNA. snORNA, the small nucleolar RNA. Both of them, you know, they they are very confusing. You have to remember snRNA is involved in mRNA processing and poly A addition during the last phase of translation, and snORNA is involved with rRNA processing, maturation, and methylation, capping and everything to stop the further hydrolysis of the RNA. Regulatory rRNA, that is siRNA and miRNA, these are involved in regulation of genetic gene expression, so regulation of transcription and translation. So this regulates gene expression, silencing of gene expression. So this is where they are called regulatory rRNA. Now let's see a question. Which of the following is not a structural characteristic of rRNA? A. Usually single stranded. Contains uracil instead of thymine. C. Contains ribose sugar. D. Forms a double helix similar to DNA. So we just discussed RNA is the secondary structure of RNA is single stranded, and it does contain uracil instead of thymine and has ribose sugar as its main sugar, instead of deoxyribose, which is present in DNA. And no, it does not form double helix structure in single stranded. So yeah, D is the correct answer. Good. Perfect. Let's go to the next question. Which feature of rRNA makes it more RNA makes it more chemically reactive than DNA? So basically, what makes DNA more stable? RNA has thymine instead of uracil. B. RNA is single stranded. C. RNA contains 2' hydroxyl group on the ribose sugar. And D. RNA forms stable double helix structure. So we are talking about what makes RNA chemically more reactive. Yes, you are correct. The presence of additional OH at 2' position of ribose sugar makes it more susceptible to hydrolysis, and hence it's compared to DNA, it's more reactive. So the correct answer is C. You can see here, even in this structure, compare deoxyribose to ribose. You can see the presence of additional OH at 2' and so this makes the ribose sugar chemically more reactive, more susceptible to hydrolysis, and hence more reactive. The next question is, which type of RNA is involved in regulation of gene expression? rRNA, tRNA, miRNA, and mRNA. So we just saw here the different types of gene. Now these are called regulatory gene. You can see here miRNA, it regulates gene expression. So just remember it now. Instead of the main things that you have to remember is tRNA, mRNA, and rRNA. All the others you can just remember because they are not involved directly in the process of transcription or translation. They have very minute minute role to play, important but minute. So it's better just to remember. The correct answer here is miRNA. microRNA. They're short and non-coding DNA that binds complementary to mRNA sequence to sub basically from. Yeah, we'll discuss that. Yeah, I think I have put it in the slide. Yeah. To suppress the translation or degrade the mRNA. Which type of RNA is directly involved in protein synthesis by carrying amino acid to the ribosome. mRNA, rRNA, tRNA, snRNA. Perfect. Yes, that is correct. The correct answer is C, transfer tRNA. It brings the set of you know codons, and it has the anticodons to it. It basically carries different codons of um, different codons for the protein synthesis. So basically, yeah, tRNA is directly involved in protein synthesis by carrying amino acid to the ribosome. Now let's briefly discuss what central dogma is. Central dogma is basically a theory, um, which uh, which states, which gives us an idea of how genetic information is transferred from DNA to RNA to protein. So um, in order for us to get RNA from DNA, there's a process called transcription that makes basically copies all the thing from the all the stored genetic information from DNA to rRNA, and the protein, RNA further translates that information of the genetic code and translates it into protein. So DNA to RNA is transcription, and from RNA to protein it's translation. RNA to DNA is reverse transcription. Now let's see what transcription is. So transcription is the process of getting mRNA from DNA. DNA has a long sequence of genes, which includes both coding and non-coding genes, non-coding sequences. The coding sequences are called exons. They're like coding, and introns are non-coding repetitive units. So this takes place in three parts: initiation, elongation, and termination. What happens is, you know, uh, the RNA polymerase. So the DNA is double stranded, right? So the double stranded, there is a promoter region that is called TATA box. So that's called TATA box. That's the promoter region, very close to the so that's where the RNA polymerase binds. The RNA polymerase binds to the promoter region. Oh, I see. I'm sorry. Is there anything? So, yeah, the initiation starts with RNA polymerase binding to the promoter region, that is TATA box. It bind and then the DNA basically opens up. You can see here the RNA synthesis begins here, and it starts forming the complementary bond to the template strand. This is the template strand, and again it forms goes from 5' direction to the 3' direction. It starts the ring starts opening, catalyzed by RNA polymerase. So RNA polymerase is a very good enzyme. It opens up and starts resealing the RNA as well. So it opens up as well as reseals the RNA, and it keeps on going, okay, in from 5' direction to the 3 dash direction, and it starts coding, you know, in this direction as well. And that's called elongation. After elongation, once it reaches the termination side, it stops. Once it reaches the terminator, binds there, and that's where it gets terminated. The single strand of RNA comes out. That RNA has both exons and introns. It's called pre-mRNA. Now, thing to remember here is both the 3 dash end and 5 dash end is capped. That's capped to protect mRNA from being hydrolyzed, or you know to basically protect the mRNA from hydrolysis by certain enzymes to keep it safe. That's why the 3 dash end, as well as the 5 dash end of mRNA is capped, methylated basically. It's called poly A tail, the terminated part, the 3 dash end part of the mRNA. Now the mRNA that we get has both exons and introns, as you can see here. After this step, then it goes on what is called splicing, where the introns are removed. This part you can see, they are removed, and then we get mRNA, like processed mRNA, which is ready to be translated. This has only the sequencing units, and that's called mRNA. Now this mRNA is ready to code for protein and ready to go to make proteins. Yeah, let's briefly discuss translation here as well. Now translation is the process from which mRNA makes protein. And the synthesis of protein takes place in ribosome. As you're well aware, a ribosome has two units, a smaller subunit and a larger subunit. The smaller subunit attaches itself to the 5' end of the RNA. You can see here. What happens is it attaches itself, and it waits for tRNA to come there. Now tRNA, along with an enzyme called amino acyl tRNA synthase, it just binds there with the amino acid. The tRNA has amino acid. It's ready there, and it just comes to the sequence of rRNA. The smaller ribosome, so smaller subunit of ribosome, keeps moving until it reaches AUG. That is the start codon, which codes for methionine. Now it moves along the RNA, and it reaches AUG, methionine, and it comes to the larger subunit. This is where they meet, smaller subunit along with the larger subunit, which has anticodon attached to it, the tRNA. Now it starts moving. Okay, now larger subunit of tRNA has three subunits: E, P, and A. Now A is where the anticodon comes. The complementary base pair starts coming. That's where the tRNA starts attaching itself from A to P to E. You can see that here. So from A to P, once the tRNA move to amino acid moves to P and starts forming the peptide one. That's called P is called peptidyl site. Now, and then it moves to E to exit. So that's how it keeps on happening: A to P to E, and then exit. It keeps on going. The ribosomal unit keeps on moving along the 5' end to 3' end, unless it reaches the codon for stop unless it reaches stop codon UAA, UAG, UGA. So they do not code for any particular amino acid. They disrupt the ribosomal, basically they disrupt the ribosomal attachment, and hence this process is stopped there. So that's like a very brief uh, summary of translation. Now let's solve some questions to uh, test our concepts. So, which enzyme is used to join DNA fragments during molecular cloning? Uh, this should be along with recombinant DNA topic, but let's try to solve it. I think you guys know the answer. So, A, restriction enzyme. No, this is molecular Caesar. So this basically cuts the DNA. So no, this is not the correct answer. B, DNA ligase. Well, C, DNA polymerase. D, reverse transcriptase. Reverse transcriptase is an enzyme that helps with reverse transcription. That is formation of DNA from RNA. So no, this is also not involved in uh, um, you know joining of DNA fragments. Is there anyone who would like to answer this question? The correct answer is ligase. It basically forms the phosphodiester bond between the sugar and phosphate, you know, to make the DNA strand. So the next question is, the process of converting DNA to mRNA is known as A, replication, B, transcription, C, translation, and D, reverse transcription. Yes, you're right. The correct answer is B, transcription. We just looked at it. Getting mRNA from DNA, it's called transcription. Let's move to the next question. What is the enzyme responsible for RNA synthesis during transcription? A, DNA polymerase, B, RNA polymerase, C, ligase, D, helicase. So we just saw in the transcription there is only one enzyme responsible that opens up the uh, DNA as well as reseals it, and helps in the elongation process. That's RNA polymerase. You can see here, this is the enzyme that's basically catalyzing this entire process. So yeah, RNA polymerase is responsible for RNA synthesis during transcription. Let's move on to the next question. Which component of transcriptional machinery recognizes the promoter in prokaryotic transcription? A, RNA polymerase 2, B, sigma factor, D, TATA binding protein, and D, enhancer sequence. So you see, this is the promoter region. How do you identify that promoter? So the process till the formation of pre-mRNA, it's same for more or less same for eukaryotic as well as prokaryotic transcription. So that's okay. I think I forgot to mention this sigma factor. So in prokaryote, sigma factor guides the polymerase RNA to promoter sequence. So you can see the sigma factor here. The yellow colored thing here is the sigma factor. It basically guides the RNA polymerase to move along. And once it leaves the, once the mRNA is formed, it leaves it, and that's where the termination takes place. Now, next question. What is the enzyme responsible for RNA synthesis during transcription? A. DNA polymerase. B. RNA polymerase. C. Ligase. And D. Helicase. DNA polymerase, ligase, and helicase, these three are not involved in the process of transcription. They're involved in a replication process. The only enzyme involved in transcription and basically catalyzing the whole process is RNA polymerase. Let's see the next question. Which component of transcriptional machinery recognizes the promoter in prokaryotic transcription? We did this. I'm sorry. It's just a. Yeah. Next question. The process of translation occurs in A. Nucleus, B. Ribosome, C. Endoplasmic reticulum, and D. Mitochondria. Yes, that's the correct answer. The translation process is a process in which they synthesize protein from mRNA, and it happens in a ribosome, as we just discussed, smaller unit and larger subunit of ribosomes. So yeah, the process of translation occurs in ribosome. Next question. What is the function of A site in the ribosome during translation? A. It holds the growing polypeptide chain. B. It binds tRNA carrying the next amino acid to be added. C. It catalyzes peptide bond formation. D. It serves as the exit site of uncharged tRNAs. Oh, so the larger subunit of tRNA has like three units. This is site A, B, A, and this is E. What they're asking, what is the function of site A in ribosome during translation? So think about it. We just discussed this in translation. Here, you can see this is the site A. Yes, you're right. The answer is B. It is called aminoacyl site, where the tRNA comes, and before before shifting to P to form the peptide bond, that's how it enters the RNA thing. So yes, site A is where the incoming charged tRNA carrying the next amino acid binds during translation. Very good. So this is the last question. Which process allows bacteria to acquire new genetic traits from the environment? A. Transduction. B. Transformation. C. Conjugation. D. Replication. We discussed replication. So, replication is basically transfer of the same genetic information from one generation to the next. They so from first generation to the next. It does not acquire new genetic traits from the environment. So this is out of the question. What about the first three options? Transduction, transformation, and conjugation. So let's discuss. So the correct answer here is transformation. Let's discuss briefly what transformation, conjugation, and transduction is. So in process of transformation, what happens is we basically uh, take foreign DNA and introduce it to the all to a new bacteria which already has a genetic information with it. Sorry. So transformation is uptake of the naked DNA from the environment, which can be introduced, which can introduce new genetic traits in the bacteria. So this can be done naturally and artificially. In natural environment, they basically fuse and have a new set of genetic information to it. Artificially, you can just do it by chemical transduction and just by viral vector. You can introduce it by phage infection, things like that, artificially. In conjugation, what happens is like the bacteria, there's a donor bacteria and a recipient bacteria. there is there is a thing called phylus comes and this phylus basically brings another bacteria closer to itself. So the one having the phylus is called a donor cell, and the different one that comes closer to it is called recipient cell. What happens is it basically brings it closer and um, attaches itself to the donor cell, and then slowly transfer its genetic information from one to another. And now both of them have the same genetic information, and that's a shared genetic information. That's called conjugation. And in process of transduction, DNA is transferred from one bacteria to another from this phage, by a different phage like bacteriophage. And what happens is, you can see here, this already has blue bacterial DNA, and this is bacteriophage. It comes there, and during the process, it incorporates its genetic information inside the uh, bacterial cell, and this bacterial cell now manufactures more infected genetic information, which then gets transferred to another slowly this bacteriophage goes again to infect other bacterial cells, and that's how this genetic information is transferred from one bacteria to the other one. So yeah, let's go to this question again. Which process allows bacteria to acquire new genetic traits from the environment? So the answer is transformation. That's it for today. If anyone has any question, you can unmute yourself and ask the question. If not, we are done for today's session. And you know, you can, if you have any question that you'd like to ask, please free feel free to ask it in the forum, mail us, or yeah, that's how you can ask different questions. So if we're done, if no one has any question, we are done for the session. You can leave now. Thank you.